Welcome all. Today we will discuss the last module of our syllabus, that is module 7, Alkaloids and Terpenoids. Two important classes of natural products which are organic compounds isolated from nature, especially plants. In today's video we will be discussing only about alkaloids. What are alkaloids? Organic nitrogenous bases, generally of plant origin, which have marked physiological actions. So as the name indicates, alkaloids are alkaline. Why? Because they contain nitrogen bases, nitrogenous heterocyclic rings. Whenever there is nitrogen, there will be lone pair and it will be a base. And they are mainly obtained from plants, just like all other natural products. And most striking feature is that they have marked physiological actions. So due to that, they are widely used in small doses in medicines and also misused as narcotics. Historically, the first alkaloid isolated was morphine, which is obtained from opium. Now, what are the important characteristics of alkaloids? As we have learned, plant origin and they are basic in nature. They have complex structure with nitrogen heterocycles, and so naturally they will be optically active. They have significant physiological activity. And regarding solubility, since they are mostly organic and non-polar, they are only partially soluble or insoluble in water but soluble in organic solvents. You can see in the picture various plant products, various alkaloids isolated from plants, various parts of plants like root, stem, leaves, seed, flower, etc. And this is one of the interesting areas of chemical research, isolating valuable alkaloids from various plants, rare plants, and investigating their medicinal properties. We have three specific examples of alkaloids to discuss, with special emphasis to their origin, structure, isolation or extraction, and finally physiological activity. So in order to understand the structure of the three examples which we have to discuss, we should be familiar with some of the nitrogen heterocyclic rings which are given in this slide. We need the knowledge of only these three rings for our purpose. The first one is pyridine. As you know, it is similar to benzene except that one carbon is replaced by nitrogen. So this is pyridine. The second one is piperidine. It is similar to pyridine. Difference is that it is completely saturated. So it is a saturated analog of pyridine. And both of them are 6-membered rings. Now the last one, this is pyrrolidine. The third one, pyrrolidine. See, if you have two pi bonds here, it is pyrrol. You might be knowing it. And if it is completely saturated, it is called pyrrolidine. So we will now move on to various examples. The first one is nicotine, the notorious nicotine, which is present in tobacco leaves. Nicotine is the common name and its systematic name is N-methyl-2-beta-pyridyl pyrrolidine. So basically it is a pyrrolidine alkaloid. Alkaloids are classified based on the structure. So the last part of the name will be the parent name. So it is a pyrrolidine alkaloid, and if you look at the name, it is N-methyl because this methyl is on nitrogen, so N-methyl. And then numbering this pyrrolidine ring, number 1 is given for nitrogen, and then 2 for this, 2 for this carbon, then 3, 4, it goes like this, 5. This is how we number this pyrrolidine ring. So on position 2 there is a substitution, that is what? Pyridyl. It's a pyridine, so we call pyridyl. And the attachment of pyridine ring is on beta carbon, that is after nitrogen we can commonly number the carbons like this. This is the first one is alpha, the second one is beta. So beta pyridyl, so substitution is on beta carbon of pyridine, so it is beta pyridyl, and that is on second carbon of pyrrolidine ring. So that is how the name. See, it is not important in learning the name, but using the name you can remember the structure. Okay, so this is the structure. You should learn, practice drawing structure. Coming to the next point, that is isolation. How we can isolate nicotine from tobacco? See, the source is tobacco plant, systematic name or botanical name is Nicotine tabacum. How it is isolated? Mainly for isolation of alkaloids we make use of solvent extraction, and also sometimes fractional distillation, fractional crystallization, etc. So what is done is, the tobacco leaves are crushed first and extracted with cold water. See, one point you should remember here is, alkaloids, as you know, are basic in nature, and in plants or in plant cells they will remain or they will exist as their salt with some acids like citric acid, oxalic acid, or they will be existing as citrate, oxalate, etc. salts in the body of plants, in the plant cells, for to be soluble in aqueous medium, because you know the plant fluid is water mainly. So to be soluble in the cell fluid, all the alkaloids need to be in ionic form. So naturally alkaloids which are basic and they are very less soluble in water will remain as some salts like citrate, oxalate, etc. in the plant cell. So in isolation what is done is, the crushed leaves is extracted with cold water to get nicotine salts in solution. That means you will be taking some vessel and take the powdered crushed leaves or powdered leaves, and then you will add cold water, okay, and stir well for extraction, and then it is acidified and extracted with ether to remove soluble hydrocarbon. So what is done is, nicotine which will remain as some salt, and we are adding cold water, so that will go into aqueous solution, right? So nicotine will be in aqueous solution, right? Nicotine salts in aqueous part, and we are adding some acid. Why? To make sure that all nicotine remain in solution as salt. That's why we are acidifying. Then extracted with ether. That is what we add now, some ether on it. We know ether is immiscible with water, so it remains as two separate layers, and you will be stirring it well. Actually this is done in what is called separating funnel. You might be knowing this kind of vessel with a stopper here. So this is done in this vessel. You will have a stopper here also, and you will shake it well. So what happens? Distribution, partition takes place. Okay, so ether is added to remove soluble hydrocarbon. So this tobacco plant will contain some unwanted hydrocarbons that will go into the ether layer. Okay, so after shaking you will pour it into the separating funnel. Aqueous layer which will be down is collected in one beaker, and ether layer can be avoided or removed. Okay, so we can remove unwanted hydrocarbons using ether extraction. Now what? Now we take the aqueous part. Okay, so this is aqueous part containing nicotine as the salt. It is now basified. That means we add some base so that nicotine will be released, because the acidic salt, to which if you add some base, it will neutralize acid and nicotine will be freed. So a base will remain free in basic medium and remain in solution in acidic medium. Remember that point. So nicotine is freed and then it is extracted using ether. Again we add ether. So now nicotine is now free. Okay, and as liquid, and then we add again ether and shake it well. So free nicotine will go into ether layer. Now again it is separated using separating funnel, and then ether layer is collected in one beaker which contain nicotine, and then ether is evaporated off. It is distilled off, and you get crude nicotine. It will not be that pure. So to purify nicotine, we repeat the process. What we do? Again we take this impure nicotine in this beaker. Of course, only very less amount of nicotine you will get after all. And then you add oxalic acid. That means you acidify. So again nicotine will go into an aqueous solution because it turns into oxalate. And then we add base, that is KOH, to neutralize the acid. Again this nicotine will be precipitated, or it will become free, and it is you can extract again using ether, and repeating this step procedure you can purify it. So this is isolation of nicotine from tobacco plants. Now what is the physiological activity? That's very important. Extremely poisonous, as you know, tobacco. In small doses it temporarily stimulates central nervous system, leading to depression. It also increases heartbeat and causes constriction of blood vessels, thereby increasing blood pressure also. In slightly higher doses it is lethal and causes respiratory paralysis and death. Long-term smokers or chain smokers develop bronchial asthma and even lung cancer. So very toxic alkaloid is nicotine. Now second one, Coniine. The systematic name is 2-propylpiperidine. So this is a piperidine alkaloid. As we have seen earlier, piperidine is this ring, piperidine. And if we number this piperidine ring, nitrogen is given number 1, position 1, and 2 for this carbon. So on second carbon we have a propyl substitution. So it is 2-propyl, or you can also call alpha propyl piperidine. Historically, this Coniine alkaloid, we can see that it is deadly poisonous, and it is believed that Greek philosopher Socrates died because he was forced to drink this extract. Okay, let us see what is the source. Hemlock herb, a kind of small plants, herbs, hemlock. Okay, and it is the scientific name is Conium maculatum. So Coniine is obtained from hemlock plants. And what is isolation? It's very easy. It is obtained mainly from seed, so it is powdered, distilled with NaOH solution. So we add NaOH solution so that this alkaloid will become free, and it is distilled, refluxed strongly, boiled, and so it will come out. This steam and this alkaloid will come out, and it is collected. Then the distillate is extracted using ether. So that vapors coming out, that is steam along with this alkaloid condensed, will be cooled, and you get the distillate, condensed liquid, to which we add now ether. So in ether, of course, alkaloid is more soluble. So this coniine will go into ether layer and then it is separated using separating funnel. Ether layer is collected and solvent ether is evaporated off. So you get the residual liquid. But it will not be very pure, and purification is done by fractional distillation. So the residual liquid is taken for fractional distillation, and the fraction collected at 166 degrees Celsius is supposed to be coniine. Now, physiological activity, very deadly poisonous, highly poisonous this one. It affects nervous system, causing gradual paralysis followed by convulsion and finally death. It causes immediate death. So in earlier time, criminals and traitors were condemned to death giving this extract of hemlock plant. And also I said, Socrates, the great Greek philosopher, is thought to have died because of poisoning with this hemlock extract. Now the last one, piperine, as the name indicated, is coming from pepper. Systematic name is quite long, you need not worry about it, but looking into the name you can understand the structure. So what is the parent name, or to which class this belongs to? It is piperidine alkaloid. So this is piperidine. And what are the substitutions? See, in piperidine ring, nitrogen is numbered 1. So 1 hyphen, then 5,1,3 benzodiazole 5-yle. What is that? See, benzodiazole is this one. This is numbered like this. Starting from this heteroatom, there is 1 oxygen, then 2, then 3 is this second oxygen, and it goes like this. Then it is 4 here, 5, 6, 7, and then the fusion points 8 and 9 here and down. Okay, so this is how it is numbered. So you can see that the positions of oxygen is 1 and 3. Okay, so it is 1,3 benzodiazole, dioxygen, 2 oxygens are there, so it is called dioxole. And what is 5-yle? That is, as we number it here, you can see that. Okay, if you number it the other way around, just take this, you can number it this way better. So 1, 2, it is 3, then 4, and this is 5. So the attached carbon is 5 of this benzodiazole ring, so that is why it is 5-yle. Okay, so now it is 1,3 benzodiazole 5-yle, that is the substitution, and the whole thing is substituted at position 5 of this chain. And now the chain is numbered like this. See, the substitution on nitrogen, you can see it is numbered from this carbonyl carbon. There is 1, then 2, 3, 4, and 5. So it is on the 5th carbon this benzodiazole ring has come. That is why 5-1,3 benzodiazole 5-yle. Then what? 1 oxo, that is keto group is named as oxo here. So 1 oxo, that is position 1. Then 2, 4 is very clear. Position 2 and 4, there is double bond. So it is 2,4 pentadienyl piperidine. So analyzing the name, we understand the structure. That is the point. Okay, now we move on to isolation. It's obtained from pepper. The systematic name is Piper nigrum. Mainly it is obtained from black pepper. So isolation is powdered black pepper is extracted using ethanol. So add ethanol and extract. Take the extract, and then the ethanol solution is then evaporated to remove ethanol so that you get a residue. And this residue will contain some polymeric resins, substance, unwanted resins. It is removed by washing with NaOH. Okay, and it is further dissolved in, again it is dissolved in hot ethanol so that you get a very saturated solution, and slow cooling will result in crystallization of piperin. So piperin is obtained by this isolation method. And finally, physiological activity. There's no toxic effect. That's a very important point. Unlike two alkaloids which we discussed, that is Coniine as well as nicotine, this piperin is having no toxic effect. Instead, it has beneficial effects. Many advantages. You know, in our daily life, pepper is used. It's an important, essential ingredient in the kitchen. So why? It enhances digestive capacity by stimulating the digestive enzymes of pancreas and reducing gastrointestinal food transit time. And so it is used in food. In all dishes we put some pepper. And also it is an inevitable part of Ayurvedic medicines, you know. Another point is that it possesses diverse pharmacological activities like analgesic property, antipyretic, anti-inflammatory, anticonvulsant, etc. And so widely used in medicines, especially Ayurvedic medicines. And also used as preservative and also used in perfumery. So that's all about alkaloids. In the next class we will discuss about terpenoids, another class of natural products. Thank you.